All right. Well, good afternoon, everyone. Thank you so much for joining us for our CTSI Distinguished Speaker series. And I realize this is the final week of our spring quarter, yet in the midst of a COVID-19 pandemic or towards the end, I think it's really an honor to introduce the national heavyweight and giant in COVID-19 research, Professor Stanley Perlman. And Dr. Perlman received his bachelor degree in physics from University of Rochester, followed by his Ph.D. in biophysics at MIT. He then pursued his medical degree from University of Miami School of Medicine. And he started his academia profession in 1983 and quickly rose to full professor, and he was a vice chair of the Department of Pediatrics at the University of Iowa. And his academic contribution has led to multiple seminal discovery from his publication. If you were to Google his Google Scholar, we will be impressed by his citation level. But during this time, I think the most important part is his contribution during this pandemic. He was the member of the Advisory Committee on Immunization Practice, a COVID-19 Vaccine Working Group. He also served as a member of DMID. Maybe Dr. Perlman could remind us what that stands, but Dr. Perlman has been advising on Safety Monitoring Committee. And also, Dr. Perlman has not been sleeping since the pandemic. He's also served as the COVID-19 Vaccine Safety Technical Subgroup, in addition to be a special government employee as a voting member for FDA in relationship to vaccines and related biological product advisory committees. Another list, he's also been serving on Lancet Commission on COVID-19 Task Force on the origin, early control of the pandemic, and One Health Solutions to future pandemic threats. While Dr. Perlman, in addition to contributing his time to addressing pandemics, his academic contribution is monumental. In addition to being member of Fellow of American Academy of Pediatrics, American Academy of Microbiology, Fellow of American Association of the Advancement of Science, a member of the American Physician. He is also the endowed chair, Mark Stinski Chair in Virology at the University of Iowa. Recently, he was awarded the Leadership in Research Award by his institution, as well as the Regents Awards for Faculty Excellence. I can continue to tell you about his academic accolades, but I think the most important part is Dr. Perlman's contribution to inspire and enrich and empower the next generation of leaders in biomedical research. He's been training numerous medical students, PhD students, as well as physician scientists who move on to be faculty, receiving federal funding, or leaders in industry. And so without further ado, let's welcome Dr. Perlman for giving us this very timely seminar. Dr. Perlman, thank you so much for joining us.
Thank you very much for the kind introduction. I'm going to share my screen now and get started. So what I'm going to do is tell you about some of our work with coronaviruses and particularly with SARS-CoV-2, the cause of COVID-19, and also some of our work with human immune responses to the virus. So my lab has been working with coronaviruses for many, many years. I think this year, I think it'll be 39 years this year. And I became interested in it for reasons that I'll go over in a minute. But for start, I'd like to start off by just describing coronaviruses in general. So I know that everybody's become much more familiar with this group of viruses. It's been, until 2003, it was a very obscure set of viruses, but that has changed. The things we know about these viruses is that they're very large, they're 31 kb in size. They're single-stranded RNA viruses, And the positive sense, 31 kb is almost the largest RNA virus. In fact, the largest or another member of the same nidovirus family. It's a virus, it's a set of viruses that infect insects in a 40 kb large. And we don't know why the smallest animal has the largest virus, but it's true. Before SARS came about, we knew about viruses that cause disease in domestic and companion animals. So some of these were respiratory disease in cows and in people as well, which I'll discuss in a minute. And to enterotropic diseases, particularly in swine, transmissible gastroenteritis virus was a big deal in swine farms, and it's been replaced by porcine epidemic diarrhea virus and porcine delta coronavirus, all of which are coronaviruses, of course. This one is interesting, the delta coronavirus, because delta coronaviruses were originally thought to only infect birds, but it crossed over to infect pigs as well. And now in the last few months, there's been a report that this same virus has been found in the blood of some patients in Haiti who had fevers. So this is still not very clear to me how this works because it wasn't found in the gut in these patients, and I would have expected it to be, but it just emphasizes something which we learned from the pandemic, which is that coronaviruses readily cross species to infect new species. And we were interested, my lab was interested in coronaviruses for so many years because of mouse hepatitis virus. And mouse hepatitis virus causes hepatitis and enteritis. And in lab facilities, mouse hepatitis virus is one of the scourges, but the strain we used only infected the brain. So we became, the studies I'll describe initially, or a little bit of them were based on that. This is an image of what one artist who gave me this picture thought coronaviruses meant. And this was a tattoo artist who in the summer of 2020, of course, by then had not been working for several weeks or months. And instead he developed this image of a coronavirus, which he gave to me, which I have on my wall. And I think summarizes the way we think about coronaviruses. So the types of coronaviruses that were present before 2002, I mentioned the human ones. These were OC43 and 229E. There was also a chicken coronavirus called infectious bronchitis virus that affected respiratory tracts. I'd mentioned MHV, and then this other one here I didn't mention. This is a feline coronavirus. Feline coronavirus is an interesting disease for several points of view. One is that it's a disease that causes pretty innocent diarrhea in cats, but occasionally in the cats, it changes to become a much more virulent disease called feline infectious peritonitis virus. This only occurs during persistence in an animal, and the changes that occur are either in the surface glycoprotein or some of the accessory proteins of the virus. And because of this, these changes, the virus now infects macrophages. And because of this change in tropism, it now causes a uniformly fatal disease. So this is a model for some human diseases, and there's actually been some therapies developed for FIPV even before the pandemic. As I mentioned, coronaviruses like to cross species, and this is some examples. The human coronavirus is 229E, which I mentioned, probably crossed into human species about 1,000 years ago, at about the same time that another human coronavirus, NL63, crossed over. And we think these came from the same host, probably from a bat virus going through maybe intermediate species or not to humans. OC43 crossed into human species about 120 years ago. This virus can also be found in cows and other animals. We don't know whether it starts in cows or started in humans, but humans can readily infect cows and cows can infect humans. The disease bovine coronavirus the virus bovine coronavirus is very similar to OC43. And in fact, about 120 years ago, there was an entity called the Russian flu, which was a severe respiratory infection. And there's speculation that this was really caused by OC43. This is not known, but of course, this would be very nice because OC43 causes the common cold now. We all hope that SARS-CoV-2 evolves to be just a pathogen that causes the common cold. SARS crossed into human populations 18 years ago and MERS 8 years ago, and I'll discuss them a little more in the rest of this talk. Now, this is the reason that I became interested in coronaviruses. This is the only slide I'll show from our work with mouse hepatitis virus. But basically, what we see here is this virus causes infection of the brain and causes infection of the myelin and the spinal cord. And if one takes a mouse that has no T or B cells, a RAG1 knockout mouse, and infects them with the mouse hepatitis virus, what one can see is huge numbers of infected cells in the spinal cord. In fact, it looks as if almost every cell in the spinal cord is infected. But when one looks at the myelin over that spinal cord, it all looks just fine. And these are the oligodendrocytes that make myelin. The myelin itself looks great. And the axons underneath the myelin, the nerve fibers, they all look fine too. Now, if one then takes these same mice and adds back T cells at 4 days after infection, now within 7 days, one sees the virus is all cleared. There's no more brown here, or very little, and the myelin is completely destroyed. And at the same time, macrophages enter the spinal cord, and the axons underneath are damaged. So this was an interesting model for studying demyelination with perhaps relevance for multiple sclerosis, but more importantly, from over the long term, it became interesting to ask the question, why is it that these viruses can infect animals, yet their clearance involves so much tissue destruction? And this is, I think, is a common theme that we've seen in COVID-19 and in other coronavirus infections. Common cold coronaviruses, I've mentioned these already, except for HKU1, which is another virus that was discovered after the SARS epidemic in 2003. They cause about 15 to 30% of common colds. And like SARS-CoV-2, or probably not like SARS-CoV-2, but they have no durable immunity. Originally, we thought SARS-CoV-2 might have immunity that wasn't durable, but more and more reports are coming out that people who either are naturally infected or vaccinated actually have immunity that lasts at least as long as we've been able to detect it, in a year or longer. These viruses cause upper respiratory tract infections, but like SARS-CoV-2, if they're going to cause a lower respiratory tract infection, the virus is preferred to cause those infections in either aged or otherwise compromised individuals. And so before I start talking about some of our human studies, what was known about immune responses to coronaviruses before the COVID-19 pandemic. And probably the best study for the common cold coronaviruses was this one in 1990, where 15 volunteers were inoculated with a 229E, and 10 of them, the 10 with lower antibody titers, became infected, and 8 of them developed colds, pointing out the lack of durability of the infection, of the protection, I mean. And then a year later, the same volunteers came back and were reinfected with 229E, and 9 became reinfected, but none developed a cold, and 6 of the 9 shed. I don't know how much they shed, but I know it was a shorter duration. So this is what we think might be an outcome of the COVID-19 pandemic, is that we are protected, but perhaps not against reinfection, but against disease. And perhaps we shed virus, but hopefully not too much. And then we have other studies that show coronaviruses infections often reoccur after 12 months. The only point of this slide I want to make is when I described feline infectious peritonitis virus, I pointed out that this was an infection that developed from feline coronavirus within persistently infected animals. In this case, macrophages became infected. And because of this, this is the only example of antibody-enhanced infection of macrophages. This is a question that keeps being raised about vaccines, whether we will do something so that we have more macrophage infection and that this will result in worse disease. In fact, I think it's really unlikely, because this only occurs in this one particular coronavirus, FIPV. And also macrophages are not productively infected with SARS-CoV-2. So it's really unlikely to occur in that setting. Then the other point on this slide is that transmissible gastroenteritis virus has disappeared. That was the original cause of diarrhea in baby pigs. And the reason is that basically a natural vaccine arose. There was this virus, porcine respiratory virus, is very similar to GGEV, but it lacks binding to receptors in the enteric tract. So pigs are naturally immunized and pass that immunization onto their, to protect their baby pigs. And the virus has basically disappeared from pig populations. So SARS-CoV-2, getting into this now, as everybody knows, is caused by, it causes COVID-19. And this is the only slide I've ever had where I have to basically update it on a daily basis. There's now 174 million cases in the world with 3.7 million deaths. And in the US, the number of deaths is almost 600,000. So what do we know about in COVID coronavirus infected humans? We know that mucosal antibody is likely important, but not well studied. And so we think that it develops in people who have been infected with SARS-CoV-2, but the amount is not well known. It's not believed to be very durable, but this is still unknown to a great extent. We know from MERS survivors that patients often had mild disease, and so the antibody levels often waned, particularly those with mild disease. Whether that's going to be true with SARS-CoV-2, there's hints with mild disease that there is waning, but I don't think we know the full picture yet. With the original SARS epidemic, we thought antibodies were short-lived, but this is probably false because subsequent studies show that antibodies can be detected for at least 12 years. And my lab working with the Zhao lab in Guangzhou found that 18 survivors of SARS, 16 had antibodies. So antibodies probably persist. These levels are not very high, but with an amnestic response on re-exposure to the virus, we suspect that they would be protected. So people often ask about the SARS-CoV-2 antibodies, the COVID-19 antibodies. And I think just to make a few points that I think are probably known to everyone in the audience, which is that these vaccines were developed very, very quickly because much of the testing was done in parallel rather than sequentially. However, the proper safety and efficacy studies were performed. They were just done more quickly. And we don't know about the durability of the response, but we know that mRNA vaccines have been amazingly protective, greater than 90%. And this is true in all populations that have been tested so far, whether it be aged populations or minority populations who had worse outcomes in the early days of COVID-19. They seem to also be efficacious in some of the patients with comorbidities. I have to say that we know from some of our studies that patients who are immunocompromised often have very, very poor responses to the vaccines. This is not a surprise. People who receive chemotherapy, one of the teachings that we always did was that they should receive vaccines before they receive chemotherapy. So, but we've measured antibodies and T-cell responses that have been basically zero in some of these patients. The adenovirus vaccines, particularly Ad26, which is licensed or has an EUA, emergency utilization authorization in this country, are less protective, but still much more protective than we had hoped for when all this vaccine work began. And then finally, vaccine hesitancy is now the major issue in obtaining widespread immunization in this country. There's not a shortage of vaccines. Okay, so how long does immunity last? This is, as I've said several times already, we really don't know, but we do see reinfection, and this is really quite rare. And the cases generally have milder disease. And to prove reinfection, of course, you need to really isolate a virus and sequence it again, unless the reinfection occurs many, many months after the initial infection. Because before then, there's some patients who seem to secrete virus for a variable period of time, even though they're not really infectious, and secrete RNA. So what we did to getting onto our work, we were interested in measuring SARS-CoV-2 T-cell responses. So we obtained peptide pools from Alex Sete and Daniela Weisskopf from the city just south of you, San Diego. And these peptides were developed by Alex and Daniela to cover all MHC class 1, class 2 epitopes, or at least a large pool of them. And so they provided pools of peptides. They were very, very helpful. And when we started our first measurements, we were interested in seeing T-cell responses in antibody-negative patients, COVID-19 survivors. And what we found is that patients who were antibody positive had a certain amount of a certain T cell response. This was done by what's called an ELISpot. So we measured the number of cells that secreted a cytokine, in this case, interferon gamma, on exposure to a peptide. If they were antibody negative, most of them also had some sort of T cell response, though much lower. It's hard to tell on this scale. And when we looked at other when we looked at other larger pools of peptides, we saw the same thing. The antibody negative patients still has some T cell responses against the epitopes in the S protein. And then the same thing was true with the antibody negative in terms of CD8 T cell epitopes. And I should say that the reasons these levels are different, and I think I misspoke, is because if somebody was naturally infected, then of course they will have a response both to the surface glycoprotein, the spike protein, as well as to epitopes outside of the spike protein. So that's why some of these numbers are higher than here. And then one of the things that we were interested in early on, which I think is probably less of an important issue now, is when people had been previously infected and then received a first dose of vaccine, they had a really strong antibody response, and that's shown here. And one can see that the response here, this patient didn't really have a zero response. This is after infection and before vaccination. The response may have been, a couple of hundred, 1 to 200, 1 to 800. But after vaccination, the response went up to 1 to 35,000. And we became interested in these patients because the second dose of vaccine is always given for the mRNA vaccines 3 or 4 weeks after the initial dose. And from an immunological point of view, this didn't make sense. So we measured the antibody responses and the T cell responses, which are shown in blue. And this is against these different pools of peptides. And what one can see is that actually the dose of antibody, after the second dose, the antibody response went down. Some of the times it went remarkably down. Here's somebody who's over 40,000, it dropped to 10,000. Well, 10,000 is still extremely high because most people who are infected usually have responses of around 1 to 1,000. And the same thing true for vaccinees. They're usually around the levels of 1 to 1,000 or less. So these are extremely high responses. This points out that doing a second vaccination after 3 weeks in this patient didn't help his antibody response at all. His T cell response, though, did improve. But these kinds of results, and others have found similar things, led to the idea that vaccination, particularly after somebody's been infected, the second dose of vaccine should be given several weeks to months after the first dose. The decision was made not to do this as a natural policy because one never really knew what the response was to the disease itself. So people who have been infected and are vaccinated still are vaccinated after 3 or 4 weeks. But whether this is best, certainly not for most people, but maybe for some. But the main point here is that there's a remarkably high response if somebody's been infected and receives a vaccine. And this is a summary of those data showing that many patients pre-vaccination, their levels are detectable. They go very high after vaccination, and after the second dose, they come down. And after they come down, I don't have those data here. Most of these dose amounts stay pretty steady for the next several weeks to months. And then the same thing with looking at T cell responses in summary data showing that after vaccination, the amount of T cell response goes up after the first vaccination, doesn't really change that much after the second vaccination, but it's much higher than you see either after the natural infection, though actually more obvious after the vaccine itself. This is really not that different from here. I guess it's statistically different, but still there's definitely a response here that's boosted, but not as appreciably as the antibody response. And so just to summarize this part of the study, we know that patients who had COVID-19 mount a virus-specific T cell and antibody response, and it's heavily boosted by the first dose of vaccine. The second dose doesn't particularly induce an enhanced T cell response and may reduce a decreased antibody response. But the other thing we've learned is that this the second vaccination may help with some of the antibody responses to some of the variants. So that's another confounder in trying to figure out what the best thing to do. So now I'm going to move on to some of our discussion of animal models, because this is really what my lab has worked on for so many years. And animal models, for those who don't think about using mice for studies, is important for evaluation of vaccines, therapeutics, and pathogenesis. And particularly, and I'll come back to this at the end, I'd like to use these mouse and other animal studies to figure out things like what's the basis for what's called long COVID patients, patients who recover from COVID-19 may not have been particularly sick, but still have ongoing symptoms, either the lungs or the brain or the heart. So what's going on there? Anosmia, which is the loss of sense of smell, cardiac disease, and then this multi-system inflammatory disease, syndrome in adults and children. In children, it's similar to a childhood disease called Kawasaki's disease. And children often do not get severe, most of the time don't get severe disease, but this disease is severe. It occurs rarely, but it's one of the main reasons that vaccination of children is proceeding onward. There's other reasons too, but it's important to prevent this severe disease in children. Kawasaki's disease tended to have cardiac sequelae. This disease seems to have less of it, but it's still a severe entity and some children die from it. So the animals, for animal models of COVID-19, several types of animals were realized to be infectable early on: hamsters, macaques, ferrets, and cats. Also, deer are infectable. All these animals develop mild disease. Dogs can be infected by SARS-CoV-2 as well. In fact, the concern is, since these animals can be infected, could they turn into a reservoir for the virus? And this is something that came out, I think, has become even more interesting recently. So this is about a year ago now, even longer. It was realized that minks in the Netherlands were becoming infected with SARS-CoV-2. And in one farm, it was noticed that people were getting sick from them, or getting infected. I don't know how sick they were getting, but minks seemed to be dying of respiratory disease, and their caretakers were getting infected. So we think what happened on these farms is that people came in with COVID-19, infected the minks, and then minks then infected people who were susceptible. And in one farm, there was a huge fraction, almost 70% of people had evidence of SARS-CoV-2 infection, probably from the minks. The farms were cold, and this kind of outbreak and spillover was also documented in other countries, including the USA. And then recently there was an outbreak in Denmark where similar events occurred. There's a lot of minks in Denmark, 15 million minks, 5 million people. And at least 200 people are infected by minks. And the mink-derived virus that infected people was not so well characterized, but it didn't spread within human populations. It certainly mutated in minks to be able to infect them a little better. And the original plan was to cull minks, but I think this has been put on hold in Denmark. One of the things that's interesting, I think, now is that when people talk about the origin of SARS-CoV-2, there's a report that just appeared in the last month that said in the animal, as people know, the argument is, where did this virus come from? In the wild animal market, or it's actually a seafood market where the amplification events seem to occur to a large extent in Wuhan. There's now a documentation that exotic animals, including minks, were found there. So if this is in fact verified, and if the minks were alive, this could well be a source for virus. There's no evidence for this. We don't have those minks. We don't know. But minks are highly susceptible to SARS-CoV-2. So to describe briefly some of the approaches that we've used to studying these the SARS-CoV-2 in mice. Mice are normally resistant to SARS-CoV-2, but it only is because there's a 1 to 2 amino acid incompatibility between mouse ACE2 and SARS-CoV-2. So there's several ways to overcome this barrier. One way which we had used in developing models for MERS was to transduce mice with adeno5 expressing human ACE2. This results in an infection that's very mild, but is easy to construct an adenovirus expressing the receptor, and it's also can be used in any mouse strain without breeding. So this is very nice in the beginning when one doesn't have other ways to study an infection. Well, we had also developed ACE2 transgenic mice for SARS, which I'll describe in a minute. We've developed ACE2 knock-in mice as well. And then finally, others, and then we did later modified the virus so it can bind to mouse ACE2. So just to tell a few things about our K18 ACE2 mice, these mice were developed probably about 15, 16 years ago now in Paul McRae's lab at the University of Iowa. Paul is my long-term collaborator. And these mice were easily infected by SARS-CoV-2. In fact, they developed an infection that was overwhelming, and it turned out to be a brain infection. So here it talks about the amount of virus needed to kill an animal as being less than 350 plaque forming units. In fact, we could show that 3 PFU was able to kill the mice, and it was all via brain disease. So when the pandemic began, we were continuing this line. We had stopped carrying it about 2009, but we were contacted by Jackson, or we contacted Jackson, I don't remember which, and we decided to resuscitate these mice. And this was done, and Jackson Labs did a terrific job in amplifying their stocks and breeding as many mice as possible. But I was worried that we were going to see a brain infection again. But in fact, it was fortunately, while the brain infection occurs, it's not as common. I don't know if I have this in the next slide. This shows some of these data. So in this first set of experiments, when we used 10 to the 5th virus, we killed all mice. But what one can see here is that 10 to the 3rd didn't. This is probably a little not quite correct. I think that 10 to the 3rd does kill some mice. And so we've considered the LD50 of this virus somewhere between 1,000 and 5,000, but a lot higher than the 3 PFU that we saw with SARS. Most important for our purposes here is the brain. These are young mice. These are 6 to 10-week-old mice. In these studies, 3 out of 6 or 3 out of 8 mice had brain disease. These 2 had low levels. These had no brain infection. So this was very different than SARS, where most of the mice, in fact, all the mice had brain disease. And this shows one of the utilities of this system is that one can see thrombi here in the lungs. And the lungs are heavily infected, but one can see thrombi here. And this is interesting because thrombi are considered one of the hallmarks of the human disease. There's definitely vascular damage in patients who have SARS-CoV-2 infection. We don't understand the etiology of it, but it seems to be more important than any other in causing disease. And in fact, in patients who came into the hospital early on, who came in with trouble oxygenating their organs, they often were put on ventilators, and this turned out to be a bad idea because the problem was these thrombi in the lungs, not necessarily the lungs themselves being damaged. So the trauma that resulted from ventilation caused more problems than the initial infection, at least initially. We saw thrombi in the liver as well. And more importantly for what I'm going to describe now, we saw thrombi in the brain. This is one of the 50% of mice that developed an overwhelming brain infection. I should also say that if one uses older mice, brain infection diminishes so that by the time mice are a few months old, there's no brain infection. And it just occurs in 50% of mice, as I said. So we became interested in in looking at another aspect of infection, namely what happens in terms of anosmia, which was just being defined as one of the features of SARS-CoV-2. And so what we did here, and we looked at the distribution of virus in the noses of these K18 mice. And what we saw was infection of the olfactory epithelium and the respiratory epithelium. Most importantly, we didn't see infection of neurons, but rather we saw infection of sustentacular cells. And these are the supporting cells in the nasal cavity in the olfactory epithelium. This is also the place where humans get infected with SARS-CoV-2. It's only the sustentacular cells, or for the most part, sustentacular cells. And these are the cells that express ACE2. And this is another way of showing that. Here, if one looks at a stain for olfactory, it's called olfactory marker protein, which marks olfactory neurons. These are in green. And this is the cell that shows, or the N protein marks infected cell. And what one can see is that the sustentacular cells are labeled while the cells in the olfactory neurons are not labeled. So we have infection of olfactory sustentacular cells, but not neurons. So so what happens in humans? So this is a human set of human nasal epithelium that we received from somebody who succumbed to the infection of 4 days after diagnosis. And one can't see it here. One can see the infection. That's these cells, these clearly labeled the magenta cells. But one can't see that they're sustentacular cells from this slide, but we think they are for where they're located. And we also saw myeloid cells located next to these infected cells. And that'll be important in a few minutes. So about the time we were starting some of these experiments, one of my students brought me this set of reviews from Amazon. And this was a reviews of scented candles. So the question was, in the reviews, do the scented candles actually work? Do they have a nice smell? Well, you can see in October and November, there's actually an increase of people who said, hey, these scented candles are not scented. And we think that's because they actually had COVID-19 as their only presenting sign. This was often the only sign of COVID-19 is the loss of sense of smell or taste. So we became interested in this with some of our sustentacular cell infection results. So at the same time, or similarly, we heard about this patient who in March of 2020, she lost her sense of smell and taste. She couldn't smell anything, food or perfume, and she recovered in about 2 weeks later. But at 8 months later, she's not completely recovered. For her, this was a financial boon because she could no longer drink very expensive wine. She couldn't tell the difference from cheap wine. So she started drinking just cheap wine. But anyway, at the same time, she became interested in knowing whether she had been infected and whether this anosmia was caused by COVID-19. So this is 8 months later. This is November. So we obtained blood and plasma from her, and we ran neutralizing antibody titers. Her titer was 1 to 48. And at the same time, we looked at T cell responses, and she had a T cell response. And this is the control Epstein-Barr virus response. So this woman had almost certainly had COVID-19 in March, and her only presenting signs were anosmia and agousia, probably just mostly anosmia. I don't know if she had agousia. So we started looking at this in mice, and these experiments were all done by Abhishek Verma, a postdoc in my lab. And what he did is he infected mice, these K18 mice, with SARS-CoV-2, and then he asked, can they tell the difference between female dander and male dander? Male mice like hanging around female dander. That's what this uninfected mouse is doing, is sniffing the female dander. But by 2 days after, or 3 days after infection, they could no longer tell the difference between male and female dander. Similarly, female mice don't care about gender differences or sex differences, but they do care about novel scents versus familiar scents. And once again, they spend their time exploring the novel scent. But by 2 and 3 days after infection, they can no longer tell the difference between novel and familiar scent. So then we did my favorite assay, you give a mouse a cookie. And what we did is we gave these mice cookies and asked if they could find them. Mice normally, particularly if they haven't eaten for a bit, really like finding cookies. This mouse, for those who know the story, did not ask for a glass of milk, for which we're very thankful. So in this test, what we can see here is that in the berry food test, the male mice could find the cookies with a certain amount of time. This is how long it took them to find it. But by the time 3 days after infection approached, they could either not find the cookie at all, or they found the cookie at much longer times. And similarly, female mice also couldn't find the cookie at 2 or 3 days after infection. In some cases, and for most mice, it took longer to find it. So this is all consistent with the idea that these mice now have anosmia. And so then this is a very short divergence to convalescent plasma. For other reasons, we decided to treat our mice with convalescent plasma, which was used earlier in the pandemic. It's not used so much now. And what we found is that if one used undiluted convalescent plasma, mice were completely protected from death, and also had much less weight loss. But the reason I show this is because the convalescent plasma here, this undiluted, completely protected against brain infection, and even dilutions of the plasma did. But more importantly, there was no change in the infection of the respiratory epithelium or the olfactory epithelium. So we next queried, was there any protection against anosmia? And what one can see here, I only show this for the female mice, is that even with convalescent plasma present, these mice at days 2, 3, and 4 post-infection could not tell the difference between the familiar and novel scents and exported them both equally. So altogether, these results say these mice do have anosmia. So then we asked, how long does it take to recover? And we can see here, just looking at the buried food test, that mice actually didn't really recover very well by 27 days. I think this may be not quite right. I think they do recover more than this. The actual histology looks pretty normal at 30 days. This is an artifact, this purple line here. But at earlier times, you can still see a little bit of destruction and some destruction here at 14 days. And when one looks in the epithelium, one can see that there's many more dividing cells in the mice that at 15 days post-infection, when presumably the olfactory epithelium is repairing, than is seen in a normal mouse. We still need to quantify these data. And looking for the presence of viral RNA and antigen, we did this by immunofluorescence. And one can see is that by day 15, there was very little evidence of continuing virus infection. And by RT-PCR, this scale here, you notice is by 3 days is up at the range of about 300. By 12 days, it's about 0.4. And at 30 days, it's even less than that, maybe at 0.1. So virus is largely cleared as the mouse recovers. We think it takes longer for anosmia to disappear. And so the next questions are, why do mice and people develop anosmia from olfactory neurons and not infected? So we knew these sustentacular cells are infected, and they express ACE2 in both humans and mice. They're important in neuron homeostasis, but they do not conduct olfactory signals. And only a small fraction of the olfactory epithelium is infected. So why is there this global loss of sense of smell? Is this because the partial sustentacular cell dysfunction had global effects, or was this immune mediated? So this was the question, and we don't have all the answers yet. But one thing we found is, and this is Abhishek did this again, looking for cilia. One can see that in the olfactory epithelium, there's a nice layer of cilia all around the olfactory epithelium. But in these mice at 6 days post-infection, the cilia are partly destroyed, and it's quite absent in quite a fraction, given that sustentacular cells were only infected in a certain number of, only a certain number of sustentacular cells were infected. So this is important because olfactory receptors are actually on the cilia. So this would explain that at least part of what we're seeing. And to confirm this, what we then did is measure olfactory receptor expression. There's at least 1,000 receptors, so we chose only 10. But what one can see here is that by 2 and 6 days post-infection, receptor levels are extremely low. So this probably explains why anosmia is present at early times. At 12 days, it's some return of the receptors. Mice still have anosmia. We think this is where we're still working. So the absence and then regain of olfactory receptor expression may explain some of the process. The other thing we were interested in, could this be immune mediated? And so we obtained RNA from the epithelium and looked for interferon and cytokine and chemokine expression. And somewhat surprisingly, both interferon beta and interferon lambda levels were very low after infection. But at the same time, CXCL10 was high, but this was not a uniform expression of pro-inflammatory molecules. IL-6 was up, but it wasn't uniform. CCL2 was increased in some mice, but not in all. ISG15, coincident with the decreased interferon lambda and beta, was also diminished in mice. So this is also something we're still trying to sort out as to what the inflammatory response actually is and how it affects the results that we're seeing in terms of anosmia. And then we didn't see an increase in inflammatory cell infiltrates. This is measuring both neutrophils here, numbers, and macrophage numbers and T cell numbers, really remarkably small difference. But what we did see, we saw there was a change in distribution, so of myeloid cells. So the green here is cells that express IBO1 and are myeloid cells. They're mostly in the basal layer. They're not really where the neurons or the sustentacular cells are. But 4 days after infection, they've migrated so that they're now adjacent to infected cells in the olfactory epithelium, which in this case is the sustentacular cells. So we think this is probably contributing to the process. But again, this is still a work in progress. So just to finish up, I want to describe another set of a couple of other sets of mouse adapted virus or other ways to probe SARS-CoV-2. So what the other way I mentioned early on is to develop mouse adapted SARS-CoV-2. Initially, this was done by Ralph Barrick by using reverse genetics and looking at the S proteins. He made 2 mutations at positions 498 and 499. Juice and Joe's group in Beijing found that they could take virus from natural virus, pass it through BALB/c mice, and actually end up with a mouse adapted virus. This was a really interesting virus because it showed, first of all, even in January, that the N501 mutation, N501Y mutation was in human stocks. This is a key mutation in the B1117 and B1351 and P1 isolates, also known as the UK, South African, and Brazilian isolates, also known now, I think, as the alpha, beta, and gamma isolates. So this was already in human stocks, but didn't become important for many months after the pandemic began. And these viruses made by both groups was very attenuated, but by passaging it through mice, the virus became more virulent. This is a well-known trick to mouse adapt viruses. We developed the N501Y mutation and passaged it through young and BALB/c mice and developed a virus that both was much more virulent, now it was lethal at about 6,000 PFUs or less, and also contained many of the mutations of interest, 417, 484, and 501Y mutations, which are found in particularly the B1351 South African variant. So we use this to infect mice. This just shows that the infection is lethal. I'm not going to spend any time on this, except to show after we purified the virus, all the isolates were basically lethal in mice. And then we wanted to repeat our anosmia experiments. And what one can see here is that there's actually much greater staining in the noses of these mice, but it's very focal. So there's no staining in many other places. So there's more staining, but it's focal, and that's shown here by immunofluorescence. And in addition, when we did the anosmia experiments, we can see that by using a sublethal dose, we could see that anosmia is apparent again in the buried food test, very clear at days 3, 8, and 14, and then resolution occurs by day 21. And when we look in the nasal cavity, the only defects we see in the olfactory epithelium are in what are called Bowman's glands, slight dilation of them, and I don't know what that means exactly. But for those who know about this, that's what we're seeing here, these dilated glands. So finally, we also developed knock-in mice, and these, I only mention this because these turn out to be very interesting. So this is modifying the mouse ACE2 so that it can support human SARS-CoV-2 replication. Turns out that this virus, these mice are also develop very mild disease now if infected by human SARS-CoV-2. But if we passage virus once again, we get a virulent virus. But what's really interesting is that this virus taken out of these human SARS-CoV, out of these mouse modified mice, that virus can now infect C57 black 6 mice, even if they don't have the modification. And then the mutations that we're finding are not the usual 498, 499, or 501 mutations. So there's another way to make these mice susceptible to SARS-CoV-2, and it's still ACE2 dependent. It's just another way of binding to the mouse ACE2. And then, so the question came up, is this unique to SARS-CoV-2? So we also did the same experiment with MERS-CoV-2. I'm not going to spend much time on MERS, except to say, since the beginning of April 2012, which is now 11 years ago, 9 years ago, sorry, there's about 2,500 cases, a very high mortality, but a very infrequent infection, maybe 2 cases a week. So we wanted to ask, do these people do these mice have anosmia? We have never heard about people having anosmia. And I've spoken to my friends in Saudi Arabia, and they don't think it's a common problem there, but I wonder if it's because there's so few people had it and many were sick. And I'm not going to go over this, but just to say that we actually found sustentacular cell infected in MERS-infected mice, and it's quite extensive, and the mice get quite significant anosmia that's shown here, in the same inability to find buried food, and here inability, these are, I think, female mice and unable to distinguish novel from familiar scent. And finally, just to show that we don't see this in all our mice, if we take mice infected with mouse hepatitis virus, we see no evidence of sustentacular infection, even though these mice develop very severe brain infection, shown in red. So just to summarize it, so we think these mice infected with SARS-CoV-2 are useful for studying anosmia. There's also been reports that they're useful for ageusia, the loss of sense of taste, but we haven't done that. Female mice develop more severe disease than males, more severe anosmia than males. And this is true in humans as well, even though COVID-19 is more severe in men. And we're still working on what the mechanism of anosmia is, whether it's because there's changes in the neuronal homeostasis or inflammation, one could see the data that we had. And so this I just covered already. And so this is what we think some of our mice will be useful for. We hope they'll be useful for studying the cardiac, neurological, kidney, and endocrine sequelae that occur after SARS-CoV-2. The role of the virus in these sequelae is unknown, since it's very, very hard to find in any virus in patients at the time of autopsy. And this is the only point, is maybe there's endothelial cell activation that's causing some of these long-term and short-term effects in people. And so we're interested in seeing what the interaction is between virus and endothelial cells is in some of our mouse models. So with that, I will stop. This is Abhishek back here, who did most of this work. And the mouse-adapted virus was developed by Roy Wang and Jian Zhang. So with that, I will stop and take any questions.
Thank you very much, Stan, for this amazing experience. So we're now open to questions for Dr. Perlman.
Let's see, there's some questions here. So the first question, there's questions both in question and answer in the chat. So I'll start with the questions and answer. So the first question is, would degrading the virus in the respiratory tract help in naturally immunizing against it? I did some successful trials of myself when I had SARS, COVID pneumonia in 2003. Well, this is sort of like the vaccine that's been inactivated. I don't think I think it's been perfected to a much greater extent in China than it had been before. So I think it has some efficacy. I think that degrading the virus and putting the respiratory tract by itself wouldn't do very much. So I would think not, and I don't know, having a successful trial on myself, but presumably, Sammy, you didn't infect yourself with SARS-CoV at the same time, but it's an interesting idea. So the next question is, does COVID-19 induce autoimmune disease and organ failure? It definitely induces organ failure, but autoimmune disease, it seems like it may induce some. That's certainly been discussed. I don't know what the state of that is, how firmly that's connected. Certainly not true in most people who survive the infection, but I think it can occur to some extent. So not a great answer. And what is the mechanism for thrombosis and how to override it? This is a key question. So there are trials to try to inhibit the complement pathway. We think that may be involved, but there's really right now no good way to override it. There was just a study showing that aspirin, which people thought might be helpful, wasn't helpful. We know that NSAIDs, non-steroidal anti-inflammatories like ibuprofen and indomethacin and then such, originally it was worried that they were going to make the disease worse, but they make the disease neither worse nor better. So we don't have a good way for overriding thrombosis. Heparin was a therapy that one could consider, but I wouldn't do it in this disease. The next question is, what is the question the research is seeking to answer? We're trying to understand, at least for the last part of the talk, I was trying to understand what the basis is for anosmia. Trying to understand why people have the loss of sense of smell and taste, and then also other sequelae. What are the implications for the possible timeline of recovering from anosmia in humans? Any thoughts on possible treatments for anosmia? Yeah, this is a very good question, because some of the anosmia manifestations, like what phantasmia or parosmia, which is not only the loss of sense of smell, but now smelling one thing and missmelling it. So one smells chocolate and thinks it's actually grass, or one smells a flower and thinks it's actually something that's foul smelling. So that the parosmia, that could either be peripheral or more likely central. Phantasmia, which is having phantom smells, is almost certainly central. So we don't understand that part of it at all. Some of the anosmia, especially the part that recovers, seems to be nose-based, but how much is involved in the actual central brain and the olfactory lobe and other connections, we don't know. It's certainly something that people are very interested in. There's some evidence of inflammation, as I said, almost no evidence for virus infection. And if it turns out that it's mostly inflammation in the nose that's causing the problem, then one might be able to easily tone down inflammation for patients who have anosmia, particularly if they have no other symptoms. But this is, again, something that's still speculative because we don't know now. Next question is, besides downregulation of olfactory receptors, is there any evidence of apoptosis? Yeah, we've been looking for that and we have not found that. We know the cells die. We have other evidence for that, but we so far have not found the mechanism of death. We know they die because we have ways to measuring surviving cells, and we know that no cells that are in the olfactory epithelium, or basically none, can be detected a week or two later after mice recover. So there's a little evidence for that, but we think it should be there. What model is best for testing therapeutics? All these are useful for therapeutics because they all allow you to infect mice, and then in some case, not all, some allow you to measure death as an endpoint or severe disease as an endpoint. Others allow you to measure levels of virus. So for example, the adenovirus system, one can use transduce mice and then measure the effect of therapies on the levels of virus left in the lungs. So all these are useful. I prefer ones that don't have the brain infection, but all these are useful both for therapeutics and vaccines. In fact, there's big trials being done, not trials, but studies being sponsored by the NIH now, asking the question, what vaccines, how, in terms of vaccine efficacy, what's the efficacy against some of the variants? And what we're doing is using a whole group of different kinds of mice, sometimes the K18 ACE2 mice, sometimes other versions of these mice. So there's lots of ways to do this. I personally would probably use the K18 mice because you can use the human strain, but I think the mouse adapted is also very useful because the fact that infection is confined to the lungs. Next question is, in mouse models, the age and sex as well as other comorbidities influence the outcome and lethality in mice from viral infection. So age certainly does. Many of our system, old mice get sick and succumb to the infection. And sex is a little different because in people, we know men do worse than women. In our mice, older males do worse than older females. But in young mice, it seems like there's not such a clear differentiation. Younger females may in fact do worse. So this is still not really clear to me. With SARS and MERS, male mice always did worse. Next question is, the anosmia tests you use are so cool. I was wondering what your thoughts were on female mice, people developing more severe anosmia than males. Is this due to the inflammatory response? Also, people looked at whether the new viral strains lead to difference in severity of anosmia. I have not seen any data about variants in anosmia. It's a really good question. In terms of the inflammatory response, I think that's really a good question. We haven't done that, but I think now that you raised that point, I think we should. I think we should look at the differences in inflammation, because maybe it would be striking. We tend to use male mice. We like to use male mice for these experiments because many of these olfactory experiments are detected by the estrus cycle in female mice, even though our effects were more prominent in female mice. So it may be a combination of inflammatory differences, endocrine differences, but these are really good questions I don't have good answers for. And in terms of viral strains, I think, some people say that up to 98% of people who have COVID-19 actually have some level of anosmia. So I don't know if that's true. We may be talking about qualitative differences or quantitative differences rather than qualitative differences, but I don't know the answer to that otherwise. Then next question is, I'm curious about those handful of antibody negative subjects you mentioned who had demonstrable T cell responses. Did you check them for monogenic antibody deficiencies, disorders? We did not. And these patients were not, these patients had moderate disease, so it wasn't like they had really terrible disease. Like, as you know, the studies that show that people who have really poor interferon responses succumb to the infection and often do very poorly. For antibody negative, we never looked. But the other thing I should say about some of these people is that what I showed probably too quickly is that if you boosted them, they actually did have T cell responses with time. So, and they did have antibody responses, but I don't know if they didn't have some relatively partial antibody defect. That's really a good question. I don't know. So that's the end of the questions. Then there's one in chat. Let's see if that's a question or not. Nope, that's not a question. So, okay, so I think I've covered all of them.
Well, thank you so much, Stan, for this very illustrious presentation. And we're looking forward to hearing more from your research. And thank you again for this amazing talk. Thank you so much.
Oh, thanks for inviting me.